Okay.
Front, back, and laser.
Okay. Thank you.
If you need any help, let me know.
All right.
Okay, I'd like to. Oops, I guess I need to
Oh,
go
you might
left.
need that.
Put this back. I'm just going to introduce Dr. Diana Six from the. She's a professor of entomology, pathology, and chair of the Department of Ecosystems and Conservation Sciences at the University of Montana.
All right. Thank you. All right. That's a little dark, but I think you can still see it. It's really nice to be here. I think it's been, I don't know, maybe 4 years since I've been to one of these things. So it's really nice to be able to come and listen to what people have been doing, kind of catch up, and also share a little bit of some of the stuff that we're doing. So today I'm going to talk about adaptation to climate change, and in particular embracing natural selection and the use of genetics in conserving whitebark pine. We hear a lot these days about resilience when we talk about forest management. Resilience is really kind of a short-term goal when we think about climate change, though, because it really strives to take a forest back to a previous condition, just allow it to do that more easily. Well, with climate change, it's not going to be so easy to go back to a current or a previous condition. We really need to start thinking more about adaptation, which of course is primarily genetic, something that will allow the trees to pretty much move forward and remain functional on the landscape. And so that's what I'm going to talk about today. And I'm going to do it in the context of mountain pine beetle, which may not surprise too many of you because that's what I work on. Mountain pine beetle is a native insect, and I think that surprises a lot of the public because the outbreak that we've seen over the last couple of decades has been so amazing that most people think that can't possibly be a native. That's got to be an exotic invader. But indeed it's a native. It can colonize most pines, not all, but most. But where you really see most of the outbreaks is lodgepole pine. Sometimes under severe drought stress, you'll see outbreaks in ponderosa. We're seeing that in California right now. And of course, we know it can happen in whitebark pine. The distribution and outbreak potential of this insect is based on a couple of things. That's the distribution of the pine hosts, but also climatic factors. And so there are some places where you're going to see lodgepoles, for instance, that you won't find mountain pine beetle. And that's because the thermal conditions are just not supportive. And you'll also find places where there's mountain pine beetle, but you don't ever see outbreak development. And again, that's all tied to thermal conditions. Now, in the last 20 years or so, a little bit longer than that, we have seen a really remarkable massive outbreak or series of outbreaks, if you want to think of it that way, across the West. If you look up here, you can see all the red. And I think you'll agree that's pretty remarkable. That's the extent of mortality. That alone is pretty amazing that we had something that big and that severe, but something that's even perhaps scarier is that with warming, this insect is expanding its range. So if you look here, the beetle used to be kind of stuck in this area right here, but in recent times it's moved across the Rockies and now has moved clear across Alberta. And in Alberta, it's now into jack pine, which is a major component of the boreal forest, and it's expected to continue to keep moving across the continent. And in that case, it would be acting as an exotic. It's moved further north. It used to stop about here, but now with warming, it's all the way to the Yukon border at this point. And of course, important to our conference here today, it has also moved upward into whitebark pine, where in many areas it has just absolutely devastated the tree. Now, one of the things that we're doing is trying not to look at these outbreaks as depressing as they do look, but use them as a learning experience. And so one of the things we're trying to do is see if we can use this outbreak as an opportunity to look for evidence that this tree has the capacity to adapt to a warming climate. One of the things we're thinking, or we are looking at, is has the outbreak resulted in kind of a big natural selection event? So in essence, in these areas that have experienced really strong outbreaks in whitebark pine, has the beetle selected trees that are not doing well, that don't have phenotypes or genotypes that allow them to deal with the warmer, drier climate? So in essence, are kind of struggling and weak, and then leave behind those that have genotypes that maybe encode for greater adaptation to drought and climate. When you walk around in these outbreak stands, at first it's kind of startling because it really does look like 100% of the mature trees have been killed. But if you walk around for a while, what you'll notice is there are trees here and there, perfectly big mature trees that, if we were, to us, they look perfectly suitable for a beetle, but they survived. And it's really kind of remarkable because if you think of what the peak populations were like during the outbreak, there were literally millions and millions of beetles that were flying. And they were beginning to run out of trees that were suitable. They were being forced into marginal trees, ones that were too small to support development. Yet, there are these big trees standing there virtually unscathed. And in most cases, if you look at them, there's not even accidental attacks. They really are virtually untouched. So one of the things we want to do is figure out what is so different about those survivor trees. And is there something about the traits that those trees have that we can help use the information to keep those trees on the landscape? Let me see. Sorry, I'm kind of sleepy. I didn't get a lot of sleep last night. It's hard to sleep in one of these things. So we're actually, with our project that we're doing right now, testing two main hypotheses. And one is that trees chosen by beetles exhibit poorer growth and defenses in recent years than survivors. So in essence, that we have trees out there in the landscape that their genotypes are such that they are not well adapted to warmer, drier conditions. They're weakened, and of course that's something the beetles like, and that they were preferentially selected. That would be incredible if we can find that. Because if we can find genotypes that are better adapted to a warmer, drier climate, that gives us something to work with. On the other hand, it may not be the case that that's happening at all. It might be that survivors merely are there because they have a very different chemical profile. Beetles find their host by sensing volatile chemicals that allow them to know, okay, this is a pine, this is a suitable pine, and that helps them locate and choose hosts. It may be that some of these trees just have chemotypes that make them either not as preferred or maybe not even recognized as a host. And that's not unusual in the plant world. In many systems, it's been found that insects avoid certain chemotypes of plants. They're actually very suitable for food, but those chemotypes just basically mask the plant there. They're not something the insect recognizes. So this is another possibility. We're doing this work in lodgepole and ponderosa pine on a much larger scale. Our work on whitebark pine is much smaller because our funding is much smaller. But we're working right now with two sites. We're looking at them very intensively to see if we can find evidence of either of these hypotheses being supported. One of the stands is Vipond Park. That one has experienced a really strong outbreak. More than 95% of the mature trees have been killed. The other one is called Comet. It's about 27 kilometers away as the crow flies. Same elevation. Both sites are nice and flat, which not only makes it nice to work, but it's important for this kind of work because that means the trees are growing under pretty similar conditions, and it's not, the effect shouldn't be due, we hope, to topography. At the outbreak stand, we've gone in and determined the size distribution of the trees that have been selected by the beetles. And then we've used that distribution to locate 100 survivors that have diameters that fall within that range. And from those trees, we measure the diameter. We take tree cores from each side of the tree to look at growth rates. We collect needles for DNA extraction, and we collect phloem to have the chemical profiles of those trees analyzed. And then we measure resin flow as a measure of their defense against the beetle. Then we located 100 beetle-killed trees, and from those we measured diameter and took tree cores. But unfortunately, we can't obviously get resin flow or resin chemistry from dead trees, nor can we get needles where we can get good DNA extractions. So in order to get a feel for what trees might be like that beetles would prefer, we actually went out and located 100 trees that are just below the diameter that the beetles would prefer. So these sort of escaped beetle selection, if you will, just by being a tad bit too small. And so from these we do all the same things we do pretty much with the live survivors, and we're hoping that that pool of samples will have genotypes both for survivors and for trees that the beetles would have selected if they were a little bigger. At the non-outbreak stand, we locate 100 live trees, and then we do all the same things we do with the survivors at Vipond. And so this is sort of an external kind of a control, if you will. So what we're doing is, one, to do tree core and climate analysis. You see some nice tree cores up here with the, hopefully you can see the blue stain. These are trees, whitebarks from the site that we cored. And we look at these, and what we'll be doing over the winter is to develop chronologies for the sites. We'll develop predictive models of tree ring width, late wood deposition, and wood density for survivors, and also trees that have been killed by the beetles. Using a couple of climate data sets, we'll calculate annual and seasonal temperature and precipitation means previously shown to affect tree growth rates. Construct model development with data from live and dead trees, and allow those responses to vary by tree status. And hopefully, when we do this, this will allow us to determine if trees that survived the recent outbreak responded to climate differently than those that were killed. I also mentioned that we're extracting DNA from needles, or I didn't, I should have. From that DNA, what we're doing is using intersimple sequence repeats. These are little chunks within microsatellite regions that are very easy and fast to resolve. They are highly polymorphic, and so with the 8 primers that we've optimized so far, we've been able to get 60 markers already. We think we'll probably have close to about 90. This is nothing like SNPs. That's the next direction we're going, but it is high enough usually to be able to resolve differences among populations. And so we're hoping that we will find our survivors segregating from the general population, and then we can screen that for actual markers that will help us detect those genotypes out in forest landscapes. We can also, of course, use correlation analysis with this ISSR data, with the phenotypic data that we're collecting on resin profiles for each tree, the resin flow, growth rates, and so forth. And so hopefully we can begin to get a feeling if these survivors are indeed different, how they're different, and perhaps markers that allow us to find them in stands where the mountain pine beetle has not yet been active. We think that these trees are not just sort of beetle resistant, but the basis for that resistance is that they actually are growing better under warmer, drier conditions. And if that's the case, then these trees may have an adaptive ability under climate change, and may give us some hope at retaining trees in areas that maybe the climate models currently are predicting that they'll completely disappear. One of the problems with climate models is that they treat trees as basically genetic clones, that there's no variation. And in fact, with trees, especially pines, they're some of the most genetically variable organisms on the planet. And so my hope is that we'll be able to detect some of the variability within this tree that we can begin to use to keep them in areas that right now we think we're actually going to completely lose the tree. We're very much, as you can tell, on the beginning stages of this. We have some indications that our hypothesis that the trees that are being selected by beetles are indeed different and that they're struggling. Connie Millar did a study in Whitebark and limber a few years ago and noticed that the trees that were picked by the beetles were growing very slowly, and those that survived were growing much faster than they did, say, 50 to 100 years ago. There was a dendrochronological study done just a few years ago, I think it was just published like 2 years ago, where they looked at whitebark pines across a whole number of sites. And they didn't really look for differences in growth rates. What they were looking for is kind of an overall signal of whether the tree would be useful in developing chronologies, but also how it's been growing over the last couple of centuries. And what they found is that across all of these sites in the West, there's been a very strong reduction in growth in general in whitebark pine, indicating it's just not really well adapted to warming and drying conditions. Our study, we're hoping, will find those individuals out there that maybe buck that trend and maybe have the genotypes and the phenotypes that allow them to exist right now, currently on some of the landscape where we think we're going to lose them. It's a kind of a collaboration right now between the University of Montana and the University of Idaho. Polly Biot is a postdoc on this with me. She's doing the modeling stuff. And I should probably switch out University of Idaho to Oregon State University because she's sort of moving. And it's funded by Montana Institute on Ecosystems and the Northwest Climate Science Center. So hopefully next year about this time, we'll have a really good update. We'll be through the genetic and the tree core analysis and the modeling and be able to share with you, hopefully, some good news. Thanks.
Thank you, Diana. We have time for a couple of questions.
Does anybody have a question? If not, I'll ask one. How do you separate out the factors of density slowing tree growth from the individual tree having some type of resistance?
We're taking stand density and basal area to every single tree that we sample. And so we're hoping that'll help us kind of factor that out. One of the things about this stand is it's both stands is that they're, it's kind of that typical really open stands, and we don't have a lot of subalpine fir and so forth. So it's a pretty open stand, but we're taking basal area and density at each tree that's sampled. Yeah.
Any other interesting questions? Yes. Of course it is.
So, question is related to population biology of what's left out there to breed. If so many of the trees die, and there's 1 or 2 trees left per acre or something, do you really have an effective population for evolution to proceed? I guess would be just a meandering thought of that.
That's a very good one. And with the data that we're gathering, we'll be able to compare genetic diversity and the effects of the outbreak on genetic diversity. And, if it's lowered substantially, even if we get genotypes that are maybe, at least right now, better adapted to warm, dry conditions, is the overall genetic diversity reduced enough that population might have a problem moving forward. We can begin to assess that with this approach. The genetic diversity thing is sort of a double-edged sword. In some cases, you would want the kind of a natural selection event to be very strong and really move the optimal fitness for that tree species more to what the current conditions are. And you don't really want a lot of other diversity because it could swamp those effects if you bring in maladapted genotypes. But on the other hand, if you reduce diversity too much, there's no material for natural selection in the future to work on. But in order to even think about that, we've got to figure out what genetic diversity exists and what's left. And so we can begin to do that.
One very quick question.
Yeah, hi. Thanks for that really interesting talk. I was just wondering if you were familiar with the studies from 2007, 2008, by Kim Wallen and Alvin Yanchuk that looked at mountain pine beetle attack resistance in progeny trials?
No, actually, I was looking at their dendrochronology work, though, but I'm not familiar with that one.
Because, yeah, they did look, because those trials have the genetic identity and relatedness of all the individuals. So they did find a bunch that had got attacked and a bunch that had escaped and did some evaluation of that. So that might be worth
Yeah, I'll have to look at that. Yeah. Those kinds of trials are really invaluable. Yeah. All right. Thank you.
Get that.